Hi everybody. We are going to be going over lab number 2, primate evolution. So now that we've worked and looked at microevolution, it's time that we explore something bigger that we know as macroevolution. And this is the evolutionary changes that create new species. And we are going to use ourselves as an example of microevolution. We'll delve into the exciting and rapid changing world of what is known as paleoanthropology. This is a study of human primate evolution. Now, we are not descended from monkeys. That is an absolutely crazy saying. And that is one of the first things people say in opposition to the theory of evolution, is that there is no way that I am a descendant of a monkey, and that's not what it's saying at all. Evolutionary theory holds that human and apes share an ancient common ancestor, and that ancestor is believed to have lived about 15 to 20 million years ago. And so I wanted to show, and throughout this lab, some pictures. Now, it's not to say, because when we go and we find these fossils, which is what we're using here to kind of uncover a pattern of evolution. And what I can say is we are looking at evidence that helps to support a hypothesized hominoid evolutionary pathway. And when we talk about looking at these evolutionary pathways, we base them on 3 types of analysis. The first thing is absolute and relative dating. Absolute dating uses the decay of radioactive isotopes, C13 or C14. This is carbon dating. Now, to let you know, we actually use other isotopes more readily because they're more stable over a longer period of time. But the carbons tend to be what we used first or used initially to determine age. But essentially, we look at how much is still left there. Because in carbon dating, if you know how that works, is there's a certain amount of radioactive carbon that living organisms have. And that produces a certain amount of radioactivity. Now, as it decays, we know how long it takes for half the amount of carbon 13 or carbon 14, how long it takes for half of it to decay. So what they can do is they can look at and quantify how much radioactivity is there and then get an idea of how old something is. So this absolute and relative dating is there. Absolute dating is there to help determine the age of fossils. Relative dating determines their age and relationships to fossils that are found in nearby either upper or lower layers of rock that they're found in. So essentially, absolute dating is actually taking the fossil itself and trying to date it. Where relative dating is, let's take the rock layers that we have and then compare it to where we find the rock or the fossil in relationship to the rock and then date the rock. Now there's molecular analysis, which is when DNA, if DNA can be harvested, and it's normally in very, very small amounts from a fossilized remain, then they can go and find out what the DNA is and then look for very specific DNA markers or sequences and compare them to different fossils or modern species. The more similar the DNA sequence is, the closer they are on the evolutionary pathway. And then a lot of what we're looking at today is morphological comparisons. Now, if you look on this picture here, there are dates on there. And they're looking at that dates using absolute and relative dating. And then they're using morphological comparisons to help kind of put them to their close relatives. And morphological comparison, this is the type of data that we're focusing on in this lab, and it refers to an organism's body shape, its bone structure, posture, wear patterns, those types of things. This data can tell us if an organism walked straight upright, which is known as bipedal, or 2-legged instead of quadrupedal, which is 4-legged. And so it can also be if it used tools, its diet, all types of things. So that's what we're going to look at when we look at this. Now, when we do something like morphological comparisons, we can essentially then put them into what we have here known as a phylogenetic tree. Now, this is a hominoid phylogenetic tree. And this is showing the descendants of our modern ancestors spread and how they diversified, and how over time it produced the modern species we find in the world today, including humans, right? And so, if you look, there's two things. If you look on page 5 of your handout, you'll notice that this phylogenetic tree exists. And I just found this from a Spanish publication that I thought was interesting, just so that you can kind of look to see what the individuals, what they believe they looked like. So if we start at the bottom, you'll see that you have this ancestral primate. And that is about 15 million years ago, and that's what we believe that gorillas, chimpanzees, and modern human beings all diverged from. Now, this is the earliest hominid lineage. This is Sahelanthropus chaadensis. And these are some $5 words, right? And so from there, you can see that it diversified. And we have Ardipithecus cadapa. And then if we go from here, this is where, if you've been to the Museum of Natural Science here, you might have seen Lucy. And this is Australopithecus afarensis. Now from here, there's a divergence. So the Paranthropus Anthiopithecus is this guy here, and essentially he diverges into two, the Robustus and Boesus. And both of these, if you see, there's nothing else that comes off of them, because that's where that lineage dies. And so if we're looking, we continue up to the right with the Homo habilis. And then you see that there is a circle that comes here. Now we have the Homo erectus, and they believe their ergaster is a very distant or close relative, or within the same class as Homo erectus. And this is the very first that they believe to use tools. And then from here, the subsection, they believe that ergaster is the one that ends up continuing on to Homo heidelbergensis. And this is where it's in here, but you have to understand that this is a developing science. They believe heidelbergensis is an ancestor to Homo sapien, but at the same point, there is some dissonance within the field that says perhaps it's not. But for the sake of here, and because it hasn't been completely disproven yet that it's not, it's very similar to the ergaster and the erectus, that they have it here. And then you can see at this point, you have the Neanderthals that are there, and Homo sapien that goes off. Now over here, I just starred them. So you can see what they believe that Sahelanthropus So over here, you can see where our earliest that we can see on here, the particular species, what they believe it looked like. And then he ends up being a ancestor of A. cadapa. And this is just kind of showing you, because I also want you to realize as you go through here, though you see these, and this is telling you millions of years here, it shows it here as well, that these species overlap. For instance, after you come through here, you'll notice that you have Lucy here which is what is right here. And this is when she's existing. And at the same time, the species that they believe she originates from this, or one of them, right? That they overlap some. So these aren't always in a vacuum. And that's why I kind of like this one over here too, because it kind of shows you that. But this is what's known as a phylogenetic tree, where it's kind of shown you, who are the ancestors, if they have where they diverge like this does, that they have a common ancestor, but there's differences. And they do this this is showing you all the fossils that they have found, specifically the skulls. And from the skulls, much like they do in forensics, when they find a body and it has been degraded down to just the skulls, and they're able to take a computer program and rebuild what that individual looks like, they do the same types of things with these fossils as they do by finding victims of violence. Now, when we look at these skulls, there are characteristics that have changed or evolved. And so if you look, for instance, the brain case. And so you can see this skull versus this one here, or even this one here, right? And you can see that the skull itself is smaller, which means what it was holding would be smaller. So the size of the brain case has allowed for a bigger brain, which in general means that there's greater intelligence, because if you look at this part in the front is where you have the frontal cortex and the prefrontal cortex, and that is what leads to higher thought. And so if that is more developed and bigger, you're going to have abstract thought happen. Now you have the changes that have occurred is the brow ridges. So you can see this is the brow ridge right here. And you can still see it in humans here. Men have a bigger brow ridge than women. And the purpose of the brow ridge is to help protect the eyes, especially if the eye sockets are more shallow. So the more shallow the eye sockets, the more pronounced the brow ridge tends to be. So women tend to have deeper, larger eye sockets than men. So that's why you'll see men with bigger brow ridges than women currently. And that's also why you'll see bigger brow ridges in our ancestors than us, because our eye sockets tend to be deeper than theirs was. And you can also look at the forehead size and the height compared to the face. Now you have in here the sagittal crest, which is also known as a cranial ridge. So if you see a presence of a sagittal crest, that means the organism has an exceptionally strong jawbone. So it produces a more powerful bite that can pierce through flesh and bone or grind down and be very, very tough through fibrous foods. And so if you look, you can see in this individual that is the sagittal crest, and you can see that it's not really as present in humans. Now the zygomatic arch, which is, you can see right here, that's what we consider. You have the zygomatic bone, which is the front part, which is kind of your cheekbones. The zygomatic arch kind of is the arching, that whole thing, there's a hole right there. So that part right there. And it's allowed for protection of the eye. And it is the origin of the masseter and part of the temporal muscle. And so it provides a connection, which is known as an articulation for the mandible. And the mandible is what we use to talk or to chew. And so the zygomatic arch, that size is different. And it makes sense because in the zygomatic arch area, which you can see here, it's large. Of course, these individuals had a more powerful bite. And we're going to also look at the characteristic changes of their teeth as well. So this is one thing that's changed. Again, like I was saying with the teeth, and there's also a change in the snout area. So we're so used to like just thinking of, you know, a snout on a pig or something. But you can kind of look at this area that's up here. And you can see that it protrudes out. And as humans are developed, it becomes more vertical. So, and snouts can have an issue because they can help to block vision below the face, making it difficult to walk upright. So if you are walking with 4, this is not a problem. But once you start walking upright, having a really large snout will make it so that you would be more likely to fall and to trip. So over time, it has reduced or gotten shorter, but that also moves the teeth more underneath. You can see how these are kind of out, and now the teeth are more in line and underneath the skull. And this gives it more flexibility in chewing and grinding food, and it allows for a more varied diet. Now, these teeth, the size of the teeth have changed. Both the molars. Molars were really, really large. They had thick enamel that are used for grinding fibrous food to crack nuts. And with this type of diet, there's not much need to cut through material with incisors or front teeth, so they tend to be smaller. So you can see we do a lot of chewing. So on ours, you can see, and this is a chimpanzee, this is us. These are some of the ancestors that we have. And on these, you can see these molars, they are bigger than humans. And we do have incisors, but their shapes are different. Now, the good thing about us, when we compare it to current chimpanzees, the amount of enamel we have on our teeth are a lot more than chimpanzees. So, you know, it allows them to be a little bit stronger. And so over time, what's happened, though, is that, you know, the canines have reduced in humans. The molars have remained large, if not gotten maybe a little bit more robust than chimpanzees. And the teeth have come and gone underneath the skull versus protruding out. And the last characteristic change that we'll talk about here is the foramen magnum, which is this hole. And so the location of it has changed. If you see, this is very much center, where this is back or towards the posterior part of the skull. And that change in location is pivotal in the change in movement. This is allowed, if you see, now that spinal column is directly centered in the skull, where before it was towards the back. So that's the difference between, you know, walking on 4 and being bipedal and walking on 2. So those are some of the changes that have evolved over time for humans. You can even see these are close. These are the ones that Neanderthals diverged off of us. And though we have similarities, you can see the difference. Look, you have the very thick brow arch. You have really thick zygomatic arches. This is kind of protruding, and it's not as protruding of a chin. Less of a forehead, so not as large of a brain with the capacity to think and complex thought. And so all of these you can see just changes even by species that would have been around at the same time. So when you're looking through and you're looking at morphological features that you're going to compare between skulls, these are the things you're looking at, right? You're going to look at the brain case, or the actual, how big is the skull? The brow ridges, the zygomatic arches, the different teeth. How far does the snout protrude? And again, you can see from the other side, you know, how deep are the eye sockets? Again, that's zygomatic bone that's here. The bone is in the front. Like I said, that when you look at the cheekbone, The arch is going to be towards the side that actually looks like it's arching out. This right here is the arch. The bone is in the front. There's actually a little line that cuts right here, and the back part is what's called a zygomatic process. That ends up making the whole arch. And then the foramen magnum. So those are the things you're going to look at as you go through this lab. Here in a minute, I'm going to tell you how you're going to do it. And I hope this is helpful for you. I really do want to make this understandable. So let's look at some of the pre-lab questions. As I always do, I'm going to go through them and help you answer some of them, and then some of them I'm going to want you to think on your own. So what is the scientific name for human? Hopefully you figure out it is Homo sapien. What science studies hominid evolution? This would be paleoanthropology. So what are the 3 types of data that we use to determine this evolutionary relationship that we're looking at? The very first thing is we have, if you remember, absolute and relative dating. We have molecular analysis. And the final thing is what we're doing with this lab, morphological comparison. Now, the rest of these, I'm going to answer one of these, but the rest of them I'm going to kind of have you think about and answer on your own. As in, are we absolutely sure the phylogenetic tree on page 5 is correct? And when you think about this, I want you to go back and think about when I mentioned earlier some of the questions that were raised, and or if there was a consensus between all scientists in this field. And that can give you an idea of what you think that answer is. Now, what you answer. What is the name of the oldest species found on the phylogenetic tree we have? Well, you could say the last common ancestor is the oldest, but we haven't found it. So it's not really the oldest species that's found on that phylogenetic tree that I showed you. It is Sahelanthropus chidensis, sorry. It's like, how many times can I say this word? Chidensis. The T is silent. And so from here, you're going to look at the pre-lab questions, and you're going to look at that phylogenetic tree, and you're going to answer the following. Approximately how many millions of years ago did humans and apes have a common ancestor? Approximately how many millions of years ago did humans and Neanderthals have a common ancestor? I can tell you out of 6 and 7, one is a lot less than the other. What does it indicate if the foramen magnum is located near the rear of the skull? Which we talked about. Where is the sagittal crest located, and how is macroevolution different than microevolution? And those you should easily be able to answer now that we finished this, and I hope that intro helped you a little bit. So now what are you going to do with the lab? Well, I've uploaded this PowerPoint that of course has all my notes in it, and you can even look, there's some additional notes underneath it. And so you're going to compare the pictures in your lab handout to the skulls in the PowerPoint. And each section is supposed to be kind of parceled out to be similar types of skulls. And so you're going to use the skulls to label the images. So you're going to go through the pictures and find out which ones look like this. And then you'd find out which one looks like this guy, and then you'd write the name here. And then you'd write a key characteristic under here. And you'll find that from the stuff that's written underneath the picture. And then you're going to answer the questions provided at the end of each one of these sections. Now, while you do this, there is a morphological comparison of primates and hominid skulls. And so you don't have to do this for every single skull. Look at the ones here, and this is telling you what table they are. So that first set you can see is here. And then you're just going to answer these questions as you go down, because it's going to allow you to compare the morphological characteristics of the different either non-ancestors or ancestors. So I'd rather you, as you do each one, as you go to that first picture, find out which one it is, and then from there, look to see if that skull is in this comparison chart and fill it out at the same time. And then once you're done, you're going to circle the names of the primates that are not involved in the evolution of hominoids or humans, and then you're going to place a star by the hominids that did not continue to evolve into modern humans, i.e. those dead-end lineages. And you'll be done with this lab. It is a bit long. It's probably going to be a bit time-consuming, so keep that in mind. You would be able to finish this probably in an hour and a half, 2 hours, and then this video will help. But this kind of shows you. So this is showing you a picture of if you were in lab, this is what you would see, right? And so these are the different, so you know there's going to be 1, 2, 3, 4, 5, right? And then what they're going to do is you're on, table 1. So this is the first one. And they're kind of showing you what they look like. And then it's going to say gorilla first. And then it's going to show you the gorilla skull. And then underneath it, you can see that it tells you it's predominantly a herbivore type of ape found in Central African forests. There's 2 distinct gorilla species. Humans and gorillas probably last had a common ancestor 7 million years ago. So you'll just write a couple things about this in that little blurb that is interesting or important for the gorilla, and then you're going to look at that, and they give you several different views. So you're going to look at that, and you're going to write. One thing I can tell you, it's got a pretty pronounced ridge. It doesn't have a very pronounced forehead. Its snout is elongated. It has huge canines. It has the sagittal ridge. Sagittal, if you know from your anatomy physiology, is just basically cutting something from right to left. So if you're cutting something straight down the body, that's sagittal, which is why it's called that. So you can label all those things. All those things we kind of talked about and had the picture about, you can write on its presence. And then on the bottom of it, showing you, look at that snout. That's a huge snout there. And then you can see where is that, foramen magnum. It's kind of towards the center. You can think about when you look at gorillas, do they always walk on all fours, or are they bipedal? So those are the questions you're going to ask. Then you're going to go on to chimpanzees. And so you're going to do this for the rest of the lab. If you have any questions, please let me know. And I hope that these videos are helpful. Have a great day.